Okay, I guess we're going to get started, try to keep on time. So now we're getting into the more advanced methods, and so now we're going to have a talk on coactivation mapping. So this is going to be Veronica Muller from the Heinrich Heine University in Düsseldorf, Germany. She'll be talking about coactivation mapping and parcellation.
All right, I now want to show you how you can use meta-analysis in order to investigate functional connectivity as well as to do coactivation-based parcellation. In the last few talks, you heard a lot about topic-based meta-analysis, where the focus of interest is mainly one specific topic or function. So in topic-based meta-analysis, we want to derive brain regions consistently found across studies investigating one specific function. For example, I'm interested in those regions which are consistently found across experiments measuring voluntary movement. So I search through the literature or through databases and identify all experiments that probe that function. I extract the coordinates that are reported in those experiments. I model the spatial uncertainty and then test it against a random spatial distribution, threshold it and correct it for multiple comparisons, and in the end I get a network which is consistently associated with voluntary movements across different experiments. However, we can also do a meta-analysis across studies testing different functions. And as was already said before, in this case, we don't identify experiments by their specific function, but we identify experiments by activation in a specific location. So in location-based meta-analysis, we want to derive those brain regions which can be consistently found to activate together with a specific seed region across studies investigating different functions. So in topic-based meta-analysis, we are just doing the analysis across experiments testing the same functions, whereas in location-based meta-analysis, we do that across experiments testing different functions. What we get out of location-based meta-analysis can then be seen as functional connectivity. Why is that so? Functional connectivity is in general defined as the temporal coincidence of spatially distinct neurophysiological events. In task-based fMRI, this can be investigated by the concurrent activation of brain regions. For example, I do an fMRI experiment and find the amygdala to be activated in an emotional task, and for the same task I found the anterior cingulate cortex to be activated. Then it can be seen as that the amygdala and the anterior cingulate cortex are co-activating. However, in location-based meta-analysis, we don't look at one single fMRI study, but we do the analysis across different experiments testing different functions and look at the co-activations consistently found. Therefore, meta-analysis can be used as a tool to derive functional connectivity, and therefore it is called meta-analytical connectivity modeling, or short MECM. As in MECM, we want to do our analysis across as many experiments as possible. Its implementation is strongly dependent on databases. As you heard in the talks before, there are a lot of open source databases which provide information about published neuroimaging papers, for example, BrainMap or the NeuroSynth database. In BrainMap, for example, there's information about around 3,000 papers which report the results of around 13,000 contrasts, which we call experiments, and those experiments report activations in around 101 different locations. Importantly, these locations are always reported in stereotactic space, and BrainMap also provides information about what was specifically done in those experiments. And that is all we need for MACM, and therefore databases are an important starting point for MACM analysis. So what are the steps, the typical steps for a MACM analysis? First of all, we identify all experiments that activate my seed region. As in topic-based meta-analysis, general and specific inclusion and exclusion criteria should be applied. As you heard in Felix' talks, general exclusion criteria means that you only include studies which report whole brain results as well as coordinates in stereotactic space. And the specific inclusion criteria means that you only include studies, for example, in healthy subjects, no group comparisons, no treatment studies. Technically, you could do an analysis across group comparisons, but you should also always be aware of what you are looking at then. For example, if you do MACM analysis across studies doing group comparisons, for example, a clinical sample to healthy subjects, then you can't interpret the results as co-activations, but rather as co-alterations. But usually in MACM, we apply general and specific inclusion criteria. We extract then all coordinates that are reported in the identified experiments and then perform a meta-analysis across those. Let's take an example. Let's assume I did an fMRI study and found the left primary motor cortex to be activated in a simple finger tapping task, and now I want to know which brain regions are functionally connected to this left primary motor cortex seed. First of all, I searched through the literature or through databases to identify all experiments that report at least one focus of activation in the seed region, so in our case, the left primary motor cortex. As I mentioned before, a good starting point for that are databases. And in our example, we used the BrainMap database and filtered it with regard to activation in the left primary motor cortex and found 155 experiments that activated. Next, we extract all coordinates that are reported in those 155 experiments from the database. And in our case, these were about 2,200 activation foci. As you can already see here visually, the foci are not evenly distributed, but cluster in some places. And for example, we find a lot of foci in the left primary motor cortex. This is not surprising because we must have at least 155 foci there because this was the criteria for experiment selection. After extracting all coordinates, we move on as in topic-based meta-analysis and perform a meta-analysis across identified experiments. Different methods can be used. In our example, we used ALE. So we extracted the coordinates and then did an activation likelihood estimation for each voxel based on the uncertainty of each foci. This map, when using ALE, can then be interpreted or seen as the probability of co-activation with the left primary motor cortex of each voxel of the brain. Then again, we test this map against the random null distribution. We threshold it and correct it for multiple comparisons, and in the end, we get a network significantly co-activating with the left primary motor cortex. As already mentioned before, we will always find the highest convergence in the seed area because this was selection criteria for the experiment, but activation outside, convergence found outside the seed area can be seen as co-activation. When comparing the results of MACAM with a topic-based meta-analysis on finger tapping as well as a single fMRI study on finger tapping, you can see that the networks look pretty similar. That means even though we are sampling across different functions in MACAM, MACAM does reveal meaningful results. When we go back to the beginning, I said that the results we find in MACAM can be seen as functional connectivity. Therefore, we can also compare our results to another functional connectivity measure, like, for example, resting state. And you can see that the network functionally connected with the left primary motor cortex seed in resting state is more extensive than in MACAM. But we also find a lot of convergence. In particular, the regions showing co-activation in MACAM, almost all of them can also be found in resting state. For example, we find the highest convergence in both approaches in the left primary motor cortex and surrounding areas. We find for both functional connectivity with the left operculum, right primary motor cortex, and SMA. But in contrast, we find also co-activations in MACAM which are not revealed by functional connectivity of resting state. Like, for example, here the right operculum insula and inferior frontal gyrus. And we also find regions functionally connected with the left primary motor cortex in resting state, but not in MACAM, like the right superior frontal gyrus. So what I wanted to show you here is that we find for both approaches to functional connectivity, one measured in the task-dependent state, whereas the other is measured in the task-independent state. We find the same networks, so convergence, but we also find some differences. In the first part of my talk, I just showed you how you can use meta-analysis to investigate functional connectivity. In the remaining time, I now go a step forward and show you how you can use information you get from MACAM in order to parcellate brain regions. So what is typically done in connectivity-based parcellation? The main aim is to identify functionally heterogeneous subclusters within a specific region of interest. That is, based on the connectivity pattern of each voxel of a ROI, we want to identify distinct clusters. So we bundle together voxels based on the connectivity profile, and the connectivity profile just means and denotes the connectivity strength of each one voxel with all other voxels of the brain. You can use different connectivity measures to do connectivity-based parcellation, like, for example, DTI, but also functional data like resting state and MACAM. Here I will only focus on MACAM connectivity-based parcellation as we are in the meta-analysis session. How does a typical MACAM CBP look like? We start with a MACAM analysis for every individual voxel of the region of interest. The important thing here is really that you don't do MACAM for the whole region, but for every individual voxel separately. This then leads to a connectivity matrix which reflects the probability of coactivation for every voxel of the region of interest with all other voxels of the brain. Next, distances between each voxel pair of the region of interest are calculated in order to quantify similarity. You can do that by, for example, using Euclidean distances. And this then leads to a similarity matrix. And in the last step, the actual clustering takes place. Here you can use different approaches for clustering, like, for example, hierarchical or k-means clustering. Let's again take an example. I here have a region in the posterior medial frontal cortex, and I want to know, are there functionally distinct subregions within this region? First of all, I start with performing a MACM analysis for every individual voxel of the posterior medial frontal cortex. This means for each voxel, I search through the database. I identify all experiments that activate that voxel. I extract all those voxels from all those experiments from the database, and then compute a cross-experiment convergence. Importantly, in contrast to the typical MACM analysis, we here stop and don't do any inference or thresholding because we want for each voxel the full connectivity profile. So, for example, we take this voxel, we identify all experiments that activate that voxel, extract the coordinates reported in those experiments, and then compute the cross-experiment convergence of coactivations. We do the same for this voxel and for all other voxels of my region of interest. Next, we calculate the distance in connectivity between each voxel pair of the posterior medial frontal cortex, which then leads to a similarity matrix which quantifies the similarity between voxel pairs. In red, red indicates high similarity, whereas blue indicates lower similarity. In the last step, we did the clustering. We here used k-means clustering, and voxels with similar coactivation patterns were grouped together, while voxels with different coactivation patterns were assigned to different clusters. In our example of the posterior medial frontal cortex, we found the parcellation into 5 distinct clusters, what you can also see here in the reordered similarity matrix. And in particular, we found a dorsal posterior cluster, here shown in green, a dorsal anterior cluster, shown in red, a dorsal middle cluster, shown in blue, a ventral anterior cluster in yellow, and a ventral posterior cluster, shown in light blue. In the last step, you can then post hoc additionally investigate the differences in connectivity patterns that drove this parcellation by looking at specific functional connectivity. And specific functional connectivity just means that one cluster shows stronger connectivity compared to all other clusters. For example, I calculate this by calculating a MACM analysis for each individual cluster, and then I contrast the connectivity pattern of one cluster with the connectivity pattern of all other clusters. And that's what we get out in our example. For example, the dorsal posterior cluster, which corresponds to the SMA, shows stronger functional connectivity compared to all other clusters to more motor regions. In contrast, the dorsal anterior region, shown in red, shows stronger functional connectivity to more frontal areas and inferior parietal cortex. To sum up, in addition to topic-based meta-analysis, where we want to identify networks associated to a specific function, we can additionally do location-based analysis, where we aim to identify networks co-activating with a specific region across different functions. I hope I could show you that meta-analytical connectivity modeling, the approach of location-based meta-analysis, offers a powerful approach to task-based functional connectivity, and that we can use information we get out of MACOM in order to do co-activation-based parcellation, which then enables to identify cortical modules in a data-driven fashion. Thank you.
Thanks, Veronica. Thanks for sticking to your time, too. So we have time for one question.
Interesting thought. I just have a question regarding, for example, you try to clusterize prefrontal medial and PMFC. So I was wondering, like, your finding is interesting, but does it have any histological or any other literature that prove we have this kind of cluster or pattern?
So you mean why we took that seed, or if the clustering makes sense?
So my question is, for example, in thalamus we can divide it to different nuclei, and different studying, different methods show that it's true. So if we do like a tractography and we show it, it's fine. Now you find some different pattern inside a PMFC. So do you have like any proof that these has any meaning or not?
For example, if you have a look, you can see that the more dorsal part so the more ventral part are regions of the cingulate cortex, where so we have a clustering first into cingulate and non-cingulate region, and then we get anterior to posterior separation for both the cingulate and non-cingulate regions. For the non-cingulate regions, it is nicely shown that the SMA and pre-SMA is separated. We have another clustering more anteriorly, which fits to more functional studies, so not anatomical studies, so functional studies, which also show that it is more anterior to posterior separation.
Thank you.
Okay, let's stop there. Let's thank Veronica again.